EM connectomics is a method that allows us to reconstruct large networks of neurons. That means we can actually reconstruct the intricate morphology of each individual neuron and the synaptic connections between all those neurons. And that is essential to understand the function of those large networks. As we learn, what changes in the brain is actually the connections between the neurons. They may appear or disappear, or they may become stronger and weaker. So it's really in the connectivity where information is stored. With this method in hand, we are now able to ask how higher brain functions emerge from the interactions between large numbers of neurons. The collaboration with the EM facility is really essential because the whole workflow of EM connectomics is really multidisciplinary, and it consists of many different steps that require very different expertise.
My role includes sample preparation, supporting people with image acquisition, and ensuring stable microscopy operations. There are three crucial steps in the workflow: sample preparation, image acquisition, and post-processing. We have assigned experts for each steps. A typical workflow includes fixation. This is an important step where you preserve the tissue. Then the sample is stained because you need contrast when you do imaging. Then the sample is embedded in plastic resin for being cut at nanometer precision. Acquisition will last many months. It's nice to do images, but you have to extract also the information that you need. The workflow for post-processing requires good experts in the field. What I found the most exciting is that my technical expertise will help uncover how the brain works.